I'm Joey Hulbert. I'm a postdoc at Washington State University. I'm based at the Puyallup Research and Extension Center, and we're really grateful because funding for this webinar series was provided by one through a gift from the Albert Victor Ravenholt Fund, and two through a small grant from Washington Department of Natural Resources. So we are really grateful and privileged to be here sharing this work. So I'm going to give a little intro to this disease that's emerged since Jacob completed his study. So we really started learning about the sooty bark disease on maples in the Northwest in 2020, and I'll share that story. And we'll talk about the study we did in Tacoma and study that we could do in other parts of the state or the region, and then how you all could get involved if you're interested in participating as community scientists. I do want to note that I am coming to you today from Tacoma. Again, really grateful to get to learn and study and research and discover and grow as a human being on the traditional homelands of the Puyallup tribe of Indians. And just acknowledge that us in Western Washington are really privileged to live and explore on these Coast Salish lands. I'm there although I'm coming to you today from Tacoma, I work in Puyallup. We have a research and extension center there, and I'm mentored by some really incredible people that focus on tree health. So Gary Chestagner, Marianne Elliott, and Todd Murray are really my key mentors there in Puyallup at the research center. Let's see. Slide stopped advancing. And as I noted earlier, we were grateful and fortunate to receive an incredible gift from the Albert Victor Ravenholt Fund, and with that, we're able to create this program called the Ravenholt Urban Forest Health Lab, and that's really what's leading or pushing this webinar series. So thank you for participating. So sooty bark disease is very similar from the outside, it looks similar to the big leaf maple decline that Jacob was talking about. Commonly, you'll see crown dieback. And so here's a bunch of photos that demonstrate some of the symptoms we see with this disease. Notably, crown dieback, generally, if I see a tree with branches that should have leaves, but it doesn't, I go and investigate further to see if there's any signs of sooty bark disease. Epicormic sprouting is another possible symptom that we see commonly when we find infected trees. And so if you see some trees that are putting shoots out from the stem in kind of weird ways, there's a possibility it's infected with this disease. This is some photos of the signs that we're seeing. So this is actually the fungus, and this is why it's called sooty bark disease, because it almost looks like the tree is burnt. The fungal mat is this crust that grows between the bark and the wood and pushes the bark off. So the bark flakes off, and underneath we see this black sooty looking fungus. So this is sooty bark disease. If you see these black patches where the bark has flaked off, that's a good sign of sooty bark disease. And note that I really emphasize the bark is flaked off. If you see black on the outside of bark, we don't typically think that's the same, or it's something different. So most of these pictures are from big leaf maple, where you see these big patches of bark flaking off, and underneath these black sooty fungal crusts, if you will. It looks a little bit different on other maple species, but we have recovered it on a lot of different maples. So this is red maple, where the bark is a lot thinner, but you still see this evidence of the kind of the signs. And this is another red maple that we saw yesterday, actually. So you just see these black patches where the bark has kind of flaked off in these puzzle piece shapes, and there's sooty fungal spore carps underneath. Sometimes the trees look completely healthy, and when you look a little closer, you'll find that one of the boles is dead. So it's interesting because sometimes we only find it on one bole. If the tree has a lot of different stems, it might only be obvious on one of the stems. So, for example, this picture on the top left, the tree looks great, but on closer inspection, it did have a dead bole. And it does seem like we really only see these signs once the wood is once that stem has died. Also interesting, we can readily find it on logs on the ground. So if you see a tree limb that has fallen off on the ground, there's a good chance you could find this black fungus, this sooty bark disease. Kind of our current understanding is that it gets into the trees, possibly through the fine twigs and from the top and grows down through the inside of the tree. So you might have an infected tree, but you won't know it until you fell the tree or until it's at a state where it's grown so far that it's started to show and flake, show at the bark or the stem. So in this case, we think it maybe started here, then grew out radially, and then by the time it grows all the way to the edge is maybe when you start to see the bark flake off, or the sooty bark. Recently, we have had this really cool project with Highline College in Des Moines, where they were going to fell a bunch of hazard trees on campus. And so they were kind enough to save cookies for us after they felled the trees. And we can go back and see how many of them have stains. And so that's been really interesting. This is a tree with sooty bark disease. You can see the flakes, and then this is the trees, after it was cut. And what's interesting is we are finding like this discoloration does show on the tree cores. So we're thinking that this could be an effective way to sample and determine if the tree has the disease prior to felling it. These are some incredible photos shared from Ryan Hulbert at City of Tacoma, where they felled a maple, and you can again see this stain kind of moving laterally through the tree and internally. And again, this is kind of what it looks like if you were to cut a cookie. So this disease is caused by a fungus called Biscognoxia. Well, it's in the group Biscognoxia, but its original name was Cryptostroma corticale. And it produces these really small ascospores. As far as we know, it reproduces asexually and produces these conidia that can spread in the wind or when the material is disturbed. On a Petri plate, it kind of looks like this. And the reason I wanted to highlight this is because there are some concerns about breathing in these spores. It's known to cause some allergic reactions in people, particularly in woodworkers working in closed environments or milling infected materials. So this photo is shared on iNaturalist. It's a great example. The individual's hands are covered in spores after they've handled the material. It seems kind of rare for us to see the spores actually released, but we're still trying to figure out like what times of year and what environmental conditions are consistent with the spore release. This is we worked with the Department of Public Health and some really great people in the city of Seattle and others from DNR to create this question and answer document with the Washington Department of Public Health. So if you do have questions about sooty bark disease and its impacts on human health, which is called maple bark disease, there's a good question and answer document online. So it's essentially the more exposed you are to this, the more hypersensitive you can become. So you can have allergic reactions. They get more severe the more frequently you work with this material. We also have some summaries of the health concerns on our website at WSU. And there's some links to some review documents looking at the health concerns. And if you have more questions about that, we do have information sessions every month as part of the Forest Health Watch. And so the next one is scheduled for May 17th. And that was a little bit longer time for questions and answers and more summaries about what we know about this disease. Also led by Washington Department of Natural Resources, Rachel Brooks, we have published a sooty bark disease diagnostic guide. And so this is online at WSU Extension, where it has pictures of what sooty bark disease is or what it looks like, and some lookalikes that are something different. Will the slide be available? Yes. I should mention that we will post the recordings of these talks on our webpage. But if you do want specific slides, maybe email me and I will I'll ask Jacob if he's willing to share, but you're certainly welcome to get ours. So sooty bark disease, that was a quick summary. I apologize that it's so brief, but I want to get through this within an hour. So I'm going to quickly go through kind of the story of the emergence of this disease in the Northwest as far as we know. And this is really important to our region because we know that nearly a quarter of the trees in our cities are maple species. And so if you look at Seattle, out of all the street trees that they've inventoried, 21% of them are Acer. Similarly with Renton and Portland and Bellevue. So it's really critical that we understand what's going on with maple because it's such an enormous part of our urban forests. And in some cases, we'll have entire communities or entire streets that are planted solely with maple. And so we really want to know, are these communities vulnerable to losing all their trees? So one thing that's interesting about this disease is we've been able to determine that it's been in the Northwest for a really long time. And we did this with by diving back into some herbarium specimens where in the early days, this gentleman named Hal Gorey had the wherewithal to submit these samples into the herbarium and correctly diagnose them as sooty bark disease in Cryptostroma corticale. So we went back into these herbarium specimens and confirmed with DNA sequencing that it's the same pathogen that we're dealing with in a lot of the rest of the state. We found, so this sample was found in Pullman, was collected in Pullman in 1968, so a really long time ago. So we know it's been in Pullman or Whitman County for a really long time. We went back to Maple Street recently, and we looked at some more maples along Maple Street actually, and found it again. So we know it's still there in Pullman. We also, this same individual who submitted this herbarium specimen to Pullman also submitted one from Centralia in 2007. So that's kind of where the story starts for us with sooty bark disease, is looking back at these herbarium specimens. But really in 2020 is when our interest started getting peaked. And that's because the city of Seattle did some incredible work working with Bartlett tree experts, where they started noting a lot of maples with the same kind of sooty symptoms, the same symptoms than these sooty bark cankers or lesions or crusts, if you will. And so Bartlett confirmed a number of different trees had the same fungus on it. And at that time, they confirmed that sooty bark disease was present, or this fungus Cryptostroma corticale was present on sycamore maple, Norway maple, bigleaf maple, horse chestnuts, Japanese maple, and even Pacific dogwood. So around that same time in 2020, there was a few different news articles that came out and a few different blog posts and some good resources. Definitely recommend following Rachel Brooks's work on this. She's definitely one of the region's experts on this disease, and she's at Washington Department of Natural Resources. But the Seattle Times published an article, and it got a few things right, and shared kind of where they're seeing this disease throughout Seattle at that time. But one thing that's not necessarily true is that it's not a newly discovered fungus. We've known this fungus for a long time. It's actually considered invasive in Europe, where it's killing a lot of sycamore maples that were planted intentionally, but not necessarily native. And so, but this is really the first emergence of it as a problem in the US. Around 2020, we were also fortunate to receive some funding from the Forest Service through an emerging pest grant to provide diagnostics to cities. So we were accepting samples and confirming whether or not it was sooty bark disease or Cryptostroma corticale with their support. And through that, we confirmed that it's kind of throughout the whole Puget Sound region. And this is as of 2021. But if we look at all the samples that we've processed at WSU between 2020 and 2020, the end of last year, we've kind of recovered it throughout the whole west side, and then again in Pullman. So it does seem like it's widespread, and there is some work coming out that's in review by Washington Department of Natural Resources, summarizing a survey of all the state parks in the state, well, in Western Washington. So stay tuned for that. But why is this disease emerging now? Our impression is that it's related to this change in the environment, because we think that this pathogen has been present in the Northwest for a really long time, and we know that our maple trees have been here for a really long time. So the way this disease triangle kind of works, or the concept, is that when you have the right host and the right pathogen or the right fungus and the right environmental conditions, then you'll get this disease. And so what's changed in the recent years? Jacob's really done a great job sharing about the changes in the kind of climate in the region. So as we kind of enter unusual global temperatures compared to when a lot of these trees have established, we begin to get really stressed out trees, but we also get really favorable growing conditions for fungi. So fungi really love wet springs and warm weather. So we think that this kind of longer, hotter droughts maybe are more favorable for the fungus. So this disease is essentially emerging as an issue now because we have better growing conditions for the fungus and more stressed out trees because of changes in our temperature and precipitation. And so with that, we wrote a proposal to the DNR through their community forestry assistance grants last year, and we were really grateful that they funded it. We knew that sooty bark disease was in Tacoma. We'd seen a number of unhealthy maples and confirmed it with some lab tests. We also know that maples were a pretty big part of our urban forests from the urban tree inventories that they have completed. And we know that there's some communities that are entirely planted with maples. And so we're concerned about the vulnerability to this disease and the possible human health impacts of that. So what we did is we kind of randomly visited areas across the urban forest of Tacoma, trying to look for tree health or measure tree health in the hot areas and cooler areas of Tacoma. And so we did this in two ways, but we tried to engage community members in this as well. So we did this in the fall before the trees all drop their leaves, and we'll be doing this again a little bit this spring in summer. So if you are interested in getting involved in this work, you can sign up as a community scientist at foresthealth.org/Tacoma. But with the help of community members and some of our technicians, we did two things. One, we sampled randomly selected maples throughout the urban tree inventory conducted by the city of Tacoma. So we randomly visited all these trees in green. And then we also randomly picked a bunch of different street blocks. So we randomly picked blocks throughout the city of Tacoma, and we went to those streets and we measured all the trees along the streets and looked for sooty bark disease. And so we want to do a lot more work with this, but generally we didn't find a relationship between heat and maple health, but I think that's largely because we didn't find a lot of maples in these hotter areas, and this excludes the trees that are completely dead. So there's some more work and investigation to be done there. So the last few minutes, I'm just going to share some opportunities to get involved in this. You don't have to be in Tacoma. There's lots of other opportunities, and we'd love to work with you. We have this program called the Forest Health Watch. You can sign up as a community scientist there, or subscribe to our newsletter, or find a lot of different guides for getting involved on this webpage. We really had the most fun so far working with the public to document where Western red cedar trees are healthy and unhealthy, and that project's ongoing. So if you're interested in other tree species like red cedar, I encourage you to jump on and get involved in that work. It's really exciting to see nearly 300 people have shared observations of healthy and unhealthy red cedar throughout kind of the whole Northwest region. But in terms of the maple health work, there's two different ways you can get involved. One, we encourage you to look into this app called iNaturalist. We use a mobile application called iNaturalist, which is a really awesome app for identifying things. You can post pictures of any organism and the community will help you identify stuff. But you can also join our projects and then add specific trees to those projects. The other way to get involved is by submitting samples to our lab. So I'm just going to I'll talk first about the iNaturalist projects, and then I'll talk about submitting samples. So we want to have a project called Sooty Bark Disease Watch, where you can add observations of where you see this disease. But we're kind of more interested in doing a similar approach to the red cedar study, where we want to know where trees are healthy and where trees are unhealthy. So we've created a Maple Health Watch project, and this includes any species of maple. And so if you join this project and add observations, there's questions about whether you saw any signs of sooty bark disease, or what were the symptoms of the health condition. Did it have canopy dieback or epicormic sprouting? Those kind of questions. And there is a guide created by a really incredible technician of ours, Fig DeWitts. So you can find this at foresthealth.org. Again, we also have created another iNaturalist project where we're interested in documenting these internal tree stains. So it'd be really neat to take pictures before and after if you're going to do any felling of a maple tree, and document if the tree looks healthy beforehand, but then you fell it and it has this stain. That's really interesting information to us, and we want to know how many of our trees may have this disease. We just don't see the signs of it yet. You're also welcome to send samples to our lab, and we can confirm or deny the presence of this fungus on these samples. So we've received a lot of different samples, particularly from cities and municipal staff, but we've also received some from community members. And those don't need to be very big. You know, a couple inches in size is all we really need. And we can do some lab tests after we extract DNA and confirm it's the same fungus or something different. So generally how that works is we get a sample, we'll check it under the microscope for kind of representative spores. We also try and isolate and grow the fungus from the tissues, and then we do this PCR where we try and amplify the DNA, and if it's the right sequence, then it will amplify and it tells us that it's the same fungus. This also allows us to build a collection of cultures from different tree hosts, and so we can use these cultures for other experiments, like intentionally infecting trees to see how it affects their health, and comparing different isolates from different hosts and things like that. So that's all I had this morning. I know we're running out of time, so the last thing I'll invite you to do is, if you have feedback about today's webinar, please share it on foresthealth.org/share. And you're welcome to contact me